Okay, so I'm going to talk a little bit about my own research, but I want to look at my research as a foundation for where I think we can go with rare isotopes. And so I mainly work on nitrogen biogeochemistry, and nitrous oxide has been the real focus of a lot of my research over the past 10 years. And like many greenhouse gases, CO2, methane, it has been rising due to human activity, and it causes about 6% of the human-induced global warming effect. And it's primarily produced from agricultural activities. So if we want to regulate this gas, mitigate it, this is where we need to focus our efforts. It's also a very elusive gas. This is showing us a record of N2O production at the Kellogg station over 1 year. And you get these high flux events where you can get 90% of your production in the space of a few days. So unless you're really on top of it, it's very difficult gas to capture, or a very difficult process to characterize. So it's also isotopically a very interesting molecule. There are two elements, but there are a total of 5 different isotopes that can combine and produce a total of 12 possible what we call isotopicules, isotopic molecules. So they range from mass 44 down to mass 48. And in green is basically the ones that we can characterize now today using a magnetic sector mass spectrometer, which we have in our lab. And newly emerging technologies is laser spectroscopy for isotopes, which we have just acquired, uses a 2-kilometer cell path, and it can now quantify the isotopes in green as well. But some of these isotopes are down at abundance of 1/millionth of mass 44 and have been very elusive except for the last 2 years. So new instruments have emerged. This is new instruments, Panorama. It's a double focusing mass spectrometer with incredible mass resolution and sensitivity, and they're now quantifying these isotopicules as well. So this again, just where I think the technology is going. Now this is just your basic diagram of the nitrogen cycle. Many of you are probably familiar with. This is sort of the diagram from an enzyme perspective and also a chemical perspective. There are 8 valence steps from the most reduced form of nitrogen, ammonium or organic matter, to the most oxidized form, nitrate. And there are microbes that basically transfer all these different pathways. There's a total of 15 different enzymes. And there are different flavors of those enzymes, but if you think about it, there's 15 enzymes that control the entire nitrogen cycle of the planet. And it is interesting in my thought that at the core of every one of these enzymes are metal cofactors. And this is then where I think we can use rare isotopes to go after and really begin to understand where these enzymes are active and when, and go after rates. So one example is, this is an example looking at nitrogen fixation in shipworms. Shipworms live, as we know, in wood. Wood has an extremely high carbon content, almost no nitrogen in wood whatsoever, and yet the organism is very enriched in N. The nitrogen has a CN of about 3 relative to the wood is probably around 200. So we've known for a long time that these shipworms have an association with nitrogen-fixing bacteria. But we don't really know exactly where they are, how active they are, and how that nitrogen is transferred from the bacteria to the shipworm. And this is an image of the gill of the shipworm acquired by a NanoSIMS instrument. So this is a secondary ion mass spectrometer, and it has the ability to raster across the surface at the cellular level and provide a 2D image of where the isotopes are distributed across the tissues. So in purple here are the bacterial sites, where the nitrogen-fixing bacteria were, and we actually are now demonstrating then that they are taking up nitrogen. And you can see that along the gill there is transfer of that nitrogen into the tissues of the shipworm. So we've never really been able to do that prior to this kinds of instrumentation. Now, nitrogen fixation involves enzymes with 3 different metal cofactors, vanadium, molybdenum, and iron. Some bacteria have 1, some have 2, some have 3, and they switch back and forth. We don't know exactly why or when they do this. And this is just another example of a NanoSIMS image. This is looking at an alyssum leaf surface. This is a plant that grows in an arid environment that sequesters nitrogen, nickel, to high concentrations, actually into the low percents. And with the NanoSIMS, you can actually image across the leaf surface. This is a scanning electron image, and we can map multiple isotopes. We're not just focusing on one. So we can do 23, we can do nickel 58, calcium 40, and so on. So this kind of technology I'm particularly excited about. I think it can help us understand the uptake and sites of metal utilization in cells, such as mentioned fixation, the use of iron, molybdenum, visualizing nutrient fluxes between plants, studying plant microbial competition, defining low active and inactive microbial populations in soils and how they're spatially organized, and potentially looking at horizontal gene transfer. Thank you.
Any questions for Nathaniel?
Quick one. So actually in subchemistry we actually use like heavy atom isotope effects, essentially look at different sort of mechanistic, to sort of rule out mechanisms. I was curious in the N2O that's produced, do you see like variations in terms of the isotopic contribution? Like, you know, you have these different isotopes, the ratios of like N14 and...
Oh, absolutely, yeah. Yeah, we actually use a parameter we call site preference. N2O is an asymmetrical molecule.
Yeah.
So the two nitrogen atoms have different bonds, and
Yeah.
therefore they acquire different isotope effects. And we measure the difference between the N15 and those two nitrogen atoms, and it turns out to be specific to whether the microbes are producing it by nitrification or by denitrification. So that's just one example.
Yeah, I used to do nitrous oxide chemistry as a graduate student. So it was fun.
Okay, thanks, Nathaniel.